appreciate the opportunity a lot, because I like the opportunity to talk about the things that I get to do, because a lot of people don't have any idea, honestly. Even when I came to the museum first as an intern, I had no idea that such active research was happening. It sort of felt like, oh, well, the museums are a place where there's just a bunch of drawers with bones in them. And but it turns out there's a lot that happens, and now I get to be a part of it. So yes, I will start sharing the presentation. Share screen. So I'm from the American Museum of Natural History. I am the laboratory manager for our microscopy and imaging facility. And in this lab, we serve all the 5 divisions of science. So that can be, invertebrate zoology, where we get bugs and sea anemones. It can be paleontology. It can be, well, astrophysics is lumped in with the physical sciences. We can't really help them because they only work on things that are figurative in space. But meteorites as well. But it's really quite cool to see different stuff every day. I mean, this week I'm doing a lot of paleontology, but last week I had some rocks, the experimental petrology that they're doing, and then tomorrow I'll actually be doing some insects, which is part of some undergraduate research, that they're helping some undergraduates get some research experience. But what's also pretty cool is that the lab is totally free to use. And it's all internal. So we do not accept projects from, paid projects from researchers from around the world. They have to have some association with the museum already, or request it. They come to look at specimens, and so mammalogy, for example, the people who study mammals will facilitate them getting CT time or microscope time, however it may be. That that's only partly a factor of how we got supported, or first created by the museum, but it's also, we are that busy. There would be no free time available. If we had 3 more systems of each, we would still be busy all the time. So maybe one day we'll be able to serve the rest of the world, or at least the rest of the tri-state area, for people who need imaging resources, because they are pretty particular. These are really high quality resources, and you can get an amazing amount of information out of them, which is why we would like to be able to open it up to more people, and smaller schools that do not have the same kind of resources available to them, because they're they have less funding, et cetera, et cetera. But one day we'll get there. There are 2 staff, myself and a coworker. My coworker just recently left, and so I'm currently the only one running it, but that's a story for another day. But what I'm trying to say is that we are here to help facilitate the work. I not only replace filaments, I schedule repairs and things like that. I do my own repairs if I need to. We train people, but we also, as Alex and I were sort of discussing, is that I often get involved in people's research as a product of the amount of effort it takes to do their work, or because they just need some help. It takes extra time to do what they're trying to accomplish. Other times I'm just interested in what they're doing. My background is human evolution. I don't get to do that very much. Now it's pretty much just sitting in front of instruments all day. But yeah, so our role here in the lab is really multifaceted. I teach classes, graduate level classes, also for undergraduates when they come over the summer. And also during the school year, they'll have some high school students and things. that will come here to the museum, and I'll introduce them because rarely do they have the other opportunity. It's not even common at a lot of universities here that the instrumentation, if it exists, it's in a geology department and it's for the, you know, the professors and the investigators. It's not the average student that gets to use it. So it's an invaluable skill to develop, and it makes you stand out. I'm here 5 days a week, but we are so busy. There are people here on the weekends, students that I've trained that can come in on the weekends and use it. So what do we have? We have our CT machine, our GE Vitamix S240. It's a dual tube system. So we can do higher power and also higher resolution scans of things that are, you know, maybe 12 inches across and slightly taller. You know, at a certain point, the size becomes your limiting factor, both in terms of what it can fit in the cabinet and also the density of it. So we can cover a really wide range of applications here in the building, but it is not sufficient. As you can imagine, some fossils would be really dense. And so more often than not, I'm turning fossils away because they are too dense rather than things that are too small and we can't get the resolution that they want. For SEM, we have a Hitachi S4700, our field emission. So for very small things, entomology uses it a lot. The person that's bugging me keeps poking in is from Earth and Planetary Sciences. So they're looking at rocks. And then we have our environmental SEM, our Zeiss Evo 60, which we purchased in an effort to further our non-destructive testing mission, which is, you know, we want to be able to return something to the collection so that people can look at it as it's supposed to be for many, many years in the future. So with the environmental, we don't have to code it, sputter coding. I don't know everybody's instrumentation or microscopy or imaging experiences, but I will talk as if you have a little bit. And if you have further questions, I will be happy to talk more. So that's our larger cabinet, well, chamber, I should say, rather than cabinet, SEM. And in addition to that, we have light microscopes, you know, our confocal laser scanning microscope. So that uses both light and lasers. And that was purchased on a grant from NASA. We originally were looking at Stardust. You know, there's a mission that went and like flew behind a comet. And so they were looking at Stardust to learn about the origins of the solar system. And then we have our light microscope, which shockingly was the absolute last addition to the lab. Up until then, all we had were fancy microscopes. We didn't have a basic light microscope. So this is our stacking microscope, which is really popular. So that's the overview of the lab. Alex, I don't know how you normally do things. Do people want to ask questions as I go or at the end?
So we'll do questions at the end, Morgan, so people can put them in the chat or raise their hand at the end, but we won't interrupt you for the time being. You carry on.
Perfect. I will move on then. So one of the projects I was on a few years ago was brought to me because it was sort of experimental. You know, I had access to a lot of literature and people who may have had some experience, and so they said, hey, we're going to try and do this. Let's see if it works. And so I used some of my personal time. Well, I don't get personal time necessarily, but I know when the CT machine is free, so I was able to throw some specimens in. But so here's the question. We said, well, there are these invertebrates, leeches, literally one of my least favorite creatures on the planet. And we want to learn more about them, but the current methodology is histological sectioning and imaging that way. I mean, it's a high quality method of doing things. People have been learning a lot for decades doing this, but what can we do to be non-destructive and potentially get more contextual information while we're leaving something intact? So we thought, well, soft tissue is obviously often not what people are trying to image. When you go to the doctors, when you broke your leg, they're trying to see the bone underneath the soft tissue. And so how do we focus on seeing the soft tissue? Well, if we can give it some dense properties in some way, that would be, you know, a way of trying to accomplish that. How can we do that? So we worked on that, you know, researching different ways of that people have been trying things, how on what scale it can work, small, medium, or large. And what if it's already dead? You know, if people can have their hearts imaged, right, by using some contrast dye, and we can see all the veins and vasculature and everything. So what can we do if there is no method of the animal itself moving the stain around? And so can we just do that without damaging it as much as possible, both either physically, you know, in terms of breaking it apart, or in any way that's irreversible? So the qualification I'm going to give here is that I would rarely consider what we actually did reversible. It can appear that things have returned to normal, but anyway. So this is what we started to do. So we thought, well, CT is the answer. We will be able to introduce a stain. So we will soak it in some sort of liquid that has heavy metal properties. So either iodine or osmium tetroxide, or a stain called phosphotungstic acid, PTA, and then CT it and hope that we get the results that we want. We don't know what kinds of tissues these stains will attach to, so what things will look like in the end. But there are also different constraints, and you know, the technology in CT is constantly improving. And so is it that we didn't have it before but we do have it now, and we just haven't tried it yet, or are we waiting for the technology to catch up with what we need? So we thought, well, let's just jump in. So the curator, the principal investigator on this, picked a specimen that he knew he had multiple specimens or samples of, different fixatives. We wanted to try and nail down, is there a best way to do it? You know, it was an experiment, and we wanted to publish and say, here's what we found was the highest quality imaging after various methods were attempted. And so in the end, we went with osmium tetroxide based on the results, which I'm going to show here. So the comparison of the 3 different stains we used are indicated on the right here. So as you can see, the stains attach to different tissues in very different ways. On the left, I think that was the PTA, if I remember correctly. Nothing is quite as bright or differentiated as the other 2. And so, it's partly a brightness issue, but you can see, compared to the one on the right, there's an enormous difference in each of them. Even the eyes are outlined compared to the iodine, which is in the middle, where they're, the anatomy of the eyes, which is the top features there, that you don't see the differentiation in the middle, you see it on the right. And all the little features in the organs on the right were all not only more clear, but also it was much noisier with the PTA. So clearly it didn't introduce the kinds of imaging improvements that we were hoping to get. And so not only with the osmium could we identify visually, but we could also segment them out. So in software, we could visualize the whole neural anatomy, which is, so you'll see the neurons passing along the anatomy all the way to the end where they're pointing to the eyes. I think they're called eye spots. So that was sort of unbelievable. I think the smaller diagram of the segmentation are the reproductive anatomy. So on the bottom, the ovaries. And yeah, so I was really surprised at the way that this turned out. I mean, so we didn't know how small a feature we would be able to identify with these stains, how much it would change the anatomy, how much it would change the shapes and the sizes of things. And ultimately, we did get some shrinkage of the specimen itself, but that didn't seem, while it did, obviously change the maybe a little bit of orientation of some of the organs and the features. Ultimately, all the information was exactly, roughly where it should be, which means we can learn even more about the general anatomy. So zooming in a little bit, the ocular, you can see here, the jaw, the J is the jaws. So there's a light difference in I don't know if you can see my mouse or not, but when you are at the edge of the jaws, there's a slight difference in the contrast. So that is where the jaw mechanism is. We've got some gastric features on the right, and then the top of it, or the sucker is here, where it would attach to you on the bottom right. So they're not pretty creatures, and I tried not to handle them if I didn't absolutely have to, but it was a fascinating process to go through, and we learned a lot about how I can apply these tools to other specimens in the future. So I'll show, this is a colored segmentation of the anatomy that they were interested in. So eye spots in green, the jaws in the magenta kind of color. I think the pale pink or the pale purple is the digestive organ. And so yeah, leeches. I didn't think I'd care all that much about them, but there we go. So then next up, this is my most recent work that's still in progress, pteropods. So these are tiny, tiny, tiny little creatures, sometimes called the butterflies of the sea, because they have little flaps of skin, or for lack of a better term, that literally kind of float them up as they travel the water column. These are less than 1 millimeter in size most of the time. So you can imagine that something that small requires high resolution for imaging. And so this was part of our question was, well, the study of these was trying to learn more about ocean acidification and what global warming is doing to our oceans. And so we wanted to take measurements to learn how their shells are changing in thickness, if they are, based on the changing chemistry of the ocean. If they're decreasing in density, they're weaker animals, which means they may not survive. And of course, as we know, the smaller creatures in the ocean are often the food. They're so plentiful that they become really important layers in the what's the phrase I'm looking at? The food tree. What is the phrase I'm looking for?
It's kind of pivotal in the food chain, really. The base
Food
source in the
chain.
food chain.
That's what I'm looking for.
Okay.
Exactly. Yes. So if the food at the bottom falls apart, figuratively and literally, then that has impacts all the way up. So these are, in the millions. These are consumed by lots of different animals. So we wanted to measure them, measure their volume, measure their thickness, and see if they're being, if the calcium carbonate shell is being weakened, then we are, there's a part of our food chain that might be at risk. So we knew there was a lot of measurements that we had to make, both of modern specimens and historical, of course. They have been around for a long time. And so we were also going to capture information about the historical properties for ones that may not have been around, or, may have preceded our global warming climate at that we're currently going through. So we needed to do a lot of measurements, a lot of scanning, a lot of analysis. And so we started to investigate these, and we're like, well, these are taking a long time. Can we possibly shorten the scans? And so it became a methodological study to say, actually, can we, with any statistical consistency? And so we started to play with our parameters. And so what you see on the right are some examples of settings that we would change. So B in the figure there is a shorter timing, 200 milliseconds. On the right, bottom right, 500 milliseconds. So we know that you're collecting more and more information with longer timing, but there is a drop off. Or at the very least, it flattens out in terms of what you're getting. If I had done a 5-second exposure, how much more information am I actually getting than 500 milliseconds or 200 milliseconds? And so we were trying to, as much as possible, remove the human impact on this. I can see some semblance of a shell in all 4 of these, but if it requires the human to do that, maybe it's not statistically significant. Maybe the computer couldn't do that, which means that we are having to spend time on each scan, and that's what we're trying to avoid here. So we're trying to change the acquisition settings to reduce the timing, the noise, which will get us, the tiniest bits of these shells if possible. And so on the bottom left, you're seeing what is the representation of the flexible area that we might have. So the bright white area is going to be the shell that we're confident about. As it gets dimmer or darker gray, it's areas that may contain shell, but they also may not. It has to be broken up into these squares. And so, could we say that the slightly darker gray is only, let's say, 25% covered by shell, or is it just too unreliable to, to be able to assign shell or not shell? This became, an important part of finding out how, essentially saying, can we repeat with multiple specimens? Can we repeat the same scans and have them be different settings, same specimens? And at what point is the measurement the same? Is it the fact that we are reducing noise? That means that each pixel or voxel is more reliably assigned what should be the correct gray value, which is to say what density it actually is. Is it shell or is it not? So these are some of the images we got from that. And on the left is the representation of our measurement studies. And so what we determined was, as you can see from the bottom graph, that there was actually a decent change in the volume measurement between 49 minutes of scan time and less, and then 50 minutes and more. So we went, and in the end, with 50 minutes because there was enough consistency. so we went with 50 minutes and the various parameters that compose that based on how consistent the measurements were and how much we were trying to get done. We were happy with the statistical continuity of that. So that's an ongoing project. We're no longer necessarily doing this particular approach to this question. Now it's mostly about the actual compositions and the measurements and what that means for climate. And actually, the woman who was running this, whose PhD project this was partly composed in, is down at Exeter at the Met office. So small world. So that's just a little mention of the kinds of things that I've been involved in myself. But here I'm going to start just talking about various projects at the museum and the imaging that people are doing, when they come here. So bugs on the right, I don't know if anybody knows, shark skin. That was a little while ago, but it's actually one of our favorite images to use. Most people have zero idea about what shark skin would look like. And so we use this a lot for, when kids come and, are getting a tour, or also some of our public programming. I ask them what they think that is, and almost nobody has any idea. So that's shark skin, if you never had the opportunity. I think the upper left is a bed bug, might be. I've sort of forgotten, but yeah. And then, what looks like it could have been like the claw of a Yeti or something, these hairy creatures. It's hard to image them sometimes because the preparation involved in reducing the artifacts that you get when you SEM on biological samples. And so this was very well done, and I think it's pretty cool. But of course, if any of you are familiar with SEM, your images are black and white. You can't necessarily get any color information unless you assign color to certain properties. And so these are examples of some of the meteorites that we have been imaging. And so they're looking at, the formation of the solar system, formation of planets, and how this, and what we can learn about that from the tiny little things that are flying around the, various, either, flying around our planet, or it's been collected from some other mission flying further out. And so they're looking at the compositions, what that means for, how various things will be conglomerates. Is that what I'm looking for? How they will collect. There's obviously a very interesting pattern on the bottom right, like almost a stratigraphy, sort of layering. What would have created that? And then trying to learn about the earliest materials in the solar system. So, to some extent, you have to make an assumption that materials and minerals that you find inside of other minerals must, within some range, be older. So how old can we, how old are some of these minerals, and what does that tell us? So, in this case, the red might be, I don't know, let's say magnesium, and then the yellow would be titanium or whatever. They assign colors, and then we'll learn about the chemistry. Generally, it's more reliable if you are comparing it to a standard of some kind. That is to say, you have something made out of magnesium, you get that signature, and then you compare it exactly to this. And so our system isn't as reliable as that, because in this, these were done on our environmental microscope. So there are whatever hairs left in the chamber and other, gases that might be left over, even though the vacuum is supposed to be very good. But it's never, 100% trustworthy. So within a certain margin of error, we can assign these and learn something about them. So I just thought these pictures are just very cool. So this is from our confocal microscope. So, these microscopes are not unlike what you could use, in your biology classes. There's a focal plane, there's a stage, we can put a slide on top, and then you can use an eyepiece to view it. This one costs a lot of money because there are lots of extra bells and whistles, like the fact that instead of light or visible light, you can use lasers and learn something about fluorescence, of the fluorescence properties of some of these animals. So famously, rocks fluoresce, but also insects and other biological creatures. So on the right is an anemone. That was an interesting project to do because they tried to sedate it essentially with some sort of anesthetic, and it just continued to move. And so there's a video somewhere of that. I'd have to try and find it from the student. And then in the middle is I think a fruit fly. And then unfortunately the bottom left, which is the organ that is most often used to identify species, which is their reproductive organs. So we have looked at a lot of bug genitalia here. Upper left is called a lace fly, and I was surprised at how very cool that is. I'd suspect it looks something like a leaf if you were to look at it in real life. So, confocal has a wide range of capabilities, but it is slightly less used than CT because there's a size range, has to fit on a slide, and also if it's not fluorescent, which is what you would be looking at, it should be somewhat transparent if you want to get 3D information from it. So, it can, the con part of confocal is that it can look through multiple slices while filtering out the unfocused light. And so you can get a 3D image to some extent, but there are a lot more limitations for this instrument. And so our light microscope, very often used by entomologists. On the upper left is an amber specimen, which is some of the coolest stuff. I mean, Jurassic Park is huge these days. And so, so I do often get excited about some of this amber stuff. Not that I believe we're going to recreate dinosaurs, but it is an unbelievable picture into the past. I mean, these creatures literally, compared with the fossilization process, which wears away everything but bone, obviously, and you can get some imprints, of the feathers, which is pretty cool. But with amber, I mean, this little creature literally just got stuck in some sap, and it's exactly soft tissue, hairs, and everything. It's sort of shocking that it survives all this time, and there's a lot of it. They go to it. I'm gonna guess that this one's from Burma. There's a lot of Burmese amber. Yeah, these things just get stuck in time pretty much, and perfectly preserved. It's amazing. So some of our CT work, various kinds of projects that have come through here. On the upper left is a shell. I thought that was a pretty cool cross section. Much larger shell. I should say this one is going to be a modern shell, and probably a minimum of 6 inches in size. On the right is a lizard of some kind with really long digits. And bottom left is a mammal. And on the bottom right was a really cool project that came through from anthropology years ago. They found a blade, I forget where, I want to say from Northern Africa. That's my vague recollection. And the sheath, which is the um, sort of cardboard looking thing that's wrapping it, was perfectly wrapped around it. But they knew that there was some, inscriptions or writing or carvings on the blade, but without removing the sheath, which would of course been very delicate by now, after all this time, was going to be impossible. And they'd rather not, again, try not to destroy things in the process of imaging it. And so this was wrapped around it, and we thought, well, CT scan it, and let's see what we can do. Unfortunately, the CT is great for samples that are less dense than metal. It is in fact made out of, a metallic box. So it became a little bit tricky to image, but that was a cool cultural anthropology project that they were doing. We don't get too many of those now, but the imagery is cool anyway. So, and then one of the really popular studies that came out was from Dr. Ed Stanley, who's now down in Florida. He was looking at lizards from the south of Africa and looking at the distribution of what you see in teal here, which is not the skin, but it was the bony armor plating that is just underneath the skin. So these are also called osteoderms. And the distribution of these is informative to their predator. So, for example, if something is running along the open grasslands, you might think, well, they'd be heavily armored because there is no protection. So it might look like the second from the right there. If something burrowed into rocks and they went, in head first, maybe all that's protected is towards the tail. Maybe they need to be light and agile to outpace their predator. So it might look more like the upper right. It was really interesting to see some of the data from these scans, because also this was a great example of why we couldn't have done any destructive sampling or imaging of these. You can't just send them to the beetle colony, which is you put a dead animal in there and then the beetles would eat away all the soft tissue, because you would lose all the information about exactly where these things were, where these osteoderms were. You need the contextual information in order to actually get the answers to your question. So CT was the answer. We did hundreds and hundreds of scans of these. Some more examples of the kinds of the varied kinds of armor that these would have, and these are all related, but adaptation is a crazy thing, right? And so, to do the bony parts of this animal is one thing, but to get soft tissue is entirely another. And so this was another scan that was done. And so he even appropriately colored all of the tissues. So I do apologize. It's a little gruesome. But you can see the muscles, you can see the tongue. You saw some veins even. It's pretty amazing. So this is, an extension of the staining that we were doing, so he would soak this animal in potentially the iodine, maybe. So something else that we learned was that the osmium works really good for the small animals because it doesn't travel very far. It can't soak in more than like a millimeter or two. And so it wouldn't work for this because not only would you need a lot and it's expensive and also quite hazardous, the iodine works a lot better for larger samples. So he would stain it, and different tissues will attract the or absorb the iodine differently, differentiating each of them visually for us. And here is another, I think, oh, good. So this is a bug now, a weevil. And so he stained this and did an enormous amount of region growing. This is Steve Davis, who's now in Japan, but he was here. You can see the brain in red. I forget which one's the tongue. I think there's like a purple bit that's the tongue. And then he just did all these slices through so you can see the features. But again, there's no way you would have gotten this quality of information without having CT'd it. You know, histology carried us for a long time, but again, to see it in 3D, to slice it at any place you want, and then to not, you know, to in any orientation. So, you know, if there's something interesting here, but also here, but you missed it, you know, it's CT is transformative, absolutely transformative. I can't sell you on it enough. So that was an example of a, I forget the species, but it's a weevil of some kind with a really long rostrum. So an example here of a mummified crocodile. I don't have a video or anything, but you know, marriage between anthropology and also herpetology. This was a pretty interesting project, but I wasn't too involved in it, but I thought it's a cool example to show. I don't think this is from Egypt or anything. I think it's from somewhere else. But that's all I had, actually. So whatever questions you might have, feel free.
Yes, thank you, Morgan. That was really interesting. I'm quite jealous of some of the stuff you get to work with. Yeah, quite fancy CT instrument as well. But
I can go back to any slides if want, you know, you want to. But
does anybody have any questions?
Did I really do exactly 45 minutes? I really didn't think I'd have 45 minutes.
Spot on.
What's the size limitation with the CT scan? I mean, how big, obviously with the light microscope, with the electron microscope, you're limited by the size of the shape, but how big can you get in the CT scanner?
So roughly, so my detector is 16 inches wide. However, that would be if the specimen was exactly backed up onto that. And so because it's a cone beam, and so you have to, you know, the closer you get to the X-ray source and further away from the detector, the smaller it has to be. So I would say roughly 12 inches wide, and you know, the height could be slightly more than that. We just might not be able to get in one shot if you just flip it over and scan the other part of it. But yeah, I think we've done some stuff that's 14, 16 inches tall. Like there's a small shark we had to flip it upside down, but yeah.
Any questions from anybody else? Okay, I have a couple. I think, do you have a question?
Yes, just first, congratulations. You have amazing specimens and very, very cool data. How do you do all the processing? So once you scan it, how do you do the brain and the tongue and all the things you were showing?
That's a great question. So there are a lot of software. Do you have experience in CT, or you know, just?
I'm more of an image analyst than a CT scanner, so.
Okay, so I'll just, you know, so there are more and more software applications out there these days that can help with this. We use a product called Volume Graphics a lot, VG Studio, and it, you know, you import your file, and then there are modules that allow you to, you know, you can even select a single gray value, and it can pick up all the matching gray values or gray values within a range, and you can segment those so you can eliminate, you know, information you don't need. For example, if a fossil is surrounded by a lot of matrix, we can get rid of the matrix and then find the dinosaur underneath. So that's an easy way to do it, you know. That's better if you you know, on something that's just bone versus the air or the plastic packaging that's around it. But when with the, like for that weevil, he actually drew out the pathway, because again, we can visually see the differences sometimes, but it is so hard to pick it apart from the pixels that are exactly next to it. The software sometimes can't do it as well as we can, which means you just got to spend time on it. So that took him weeks to work on. Not that he's working on it, 8 hours a day, but it's, a lot of effort. But there are other freeware. We pay volume graphics is really expensive, but there are even freeware softwares that allow you to do it. And then there are software that you can upload it, and then you can manipulate your specimen in, like, by using the VR glasses, and yeah, it's a little wild.
Thank you.
Thank you. That was actually going to be my question. So I was just about to ask about segmentation and how you achieved it. So there you go. So we use Dragonfly, which is a free software, quite a bit.
Yeah, I'm trying to use that more and more because it's really powerful now. I started to use it several years ago, but there's some modules missing that would have made it really great for us. And now it has like the bone analysis and things, and this machine learning, which is transformative, really. So it's hard to just sit down and say, all right, I'm not going to do anything else. I'm just going to sit at this computer and train myself on a new software. It takes ages.
Yes, I know that pain. Dragonfly is a really good package, and the latest iteration of it has added a lot. So yeah, that's cool. The other question that I was going to ask was about kind of correlatives. You've got so many different aspects that you use and so many different techniques you use. There's a real push for Clem and that kind of thing at the moment to push things towards a more correlative aspect. Do you find yourselves using kind of multiple instruments to answer a question very often, or?
Not often enough. Some things that would work really well would be on the meteorites. For example, we will CT scan them. They will find out where their grain or whatever feature of interest is and locate it exactly, go downstairs, cut it up, and then send it to the SEM to do full chemistry on that. Otherwise, we'll do light microscopy sometimes on something so that they can get, just accompanying information and complementary for publications. That kind of thing. You can't publish these days without some good pictures. So we're getting more and more popular.
Yes. We have a system which in theory means we can go from a confocal to a FibSem, so we can go.
Man, you know what? When I go to the Zeiss meetings and I see the things that they can do, like they had this example of a CT that they did, or X-ray microscopy, and then the software just zooms in on a ROI scan that was also done on the Versa, so it gets increasingly high resolution, and then they threw it in whatever kind of SEM, and it's all layered. I mean, it's unbelievable what people can do in terms of mixing your modalities and combining information. As soon as I have funding for that, I'll do it.
I guess that's kind of the point of events like this. Is in theory we can work together and help other different institutions help each other out with the different equipment we've got. Kind of try and foster some better interaction, because it's not necessarily always as good as it could be, is it? But there we go. Anyway, any more questions from anybody? So I don't have all the time.
Anybody, next time you're in New York, you can come and visit.
I might take it up on that.
Please do.
Okay, right. If nobody has any more questions, I'm going to thank Morgan, because that was great talk. I really enjoyed that.
Thank you.
Thank you to everybody who came up. We will put the recording up for those who weren't able to make it. I know there were quite a few who were asking for it. So the links will be sent out, and then on to the next session, which I think is going to be in marine science for the next one. So I'll keep you posted. So thank you everybody for attending.
Thank you for the invitation. It was very cool.
You're very welcome. Very welcome.
Thank
Have a good day.
you. Bye now.
Bye-bye.